Hi, this is M. Prasad Nair. Today we are discuss about the recombinant DNA technology. So it is like based like a gene recombination. So definition like it has a number of experimental protocol leading to transfer the genetic information of DNA from one organism to another. So it is involving of manipulations of genetic material of DNA achieve the desired goal in the predetermined way. The present day recombinant DNA technology has its roots in the experiments performed by Boyer and Cohen. The basic principle of recombinant DNA technology, it is generation of DNA fragment selections of the desired pieces of DNA. The insertion of the selected DNA into cloning vector creates recombinant DNA or chimeric DNA. The introductions of recombinant vector into the host cell, the multiplications of selections of clones containing the recombinant molecules, expressions of gene to produce the desired products. This is how the recombinant, this is the probe and primary acid, this is the one fragment and this is another fragment. This is the DNA and B in E. coli. Recombinant DNA, here it is new will be synthesized in joined with by the pairing of this and this to combine with this to form a recombinant DNA. This is how the whole metabolism is the pathway is going plasmid, how it will be injected, restricted enzymes is added, black plasmid, that insulin is producing. The restriction endonucleases, the enzymes for multiplications of DNA, are bacterial enzymes that can split DNA at species sites. These were first discovered in E. coli restriction of the replications of bacteriophage by cutting the viral DNA. The host E. coli DNA is protected from the cleavage by the addition of methyl groups. Those the enzymes that restricted the viral replication are known as restriction enzymes or restriction endonucleases. The recognition sequence is the site where the DNA is cut by a restriction endonuclease. Restriction endonuclease can specifically recognize the DNA with a particularly sequence 4 to 8 nucleotides and cleavage. The cleavage pattern and DNA fragments. This is the restriction endonuclease type 1 and type 2, type 3. These are the three mechanisms. Restriction endonuclease, BAM-He, coli, and He, and HPA. This is the macroorganism species. This is how enzymes, restriction enzymes. This is the restriction site that will be cutting or splitting. This is the E. coli, how this will be cutting restriction of E. coli. This is E. coli site and stick with the two strands and SMA. Restriction endonuclease producing the blunt ends and sticky ends. The DNA ligase to attach with the two molecules. These were the original isolated virus and also occurred in the E. coli Eukaryotic. The cut DNA fragments are covalently joined together by the DNA ligase. So DNA ligase the DNA fragments by forming a phosphodiesterase bond between the phosphate group 5' carbon of one deoxyribose with the hydroxyl group of 3' carbon of another deoxyribose. The host are leaving the system of cells in the carrier of recombinant DNA molecules or vector can be propagated. The host cell can be prokaryotics or eukaryotics. Microorganisms are preferred as a host cell since they multiply faster compared to the cells of higher the organism. E. coli experiments. This is plasmid insertion. How this will structure. So eukaryotic hosts, these are the preferred to produce the human proteins since they have complex structures suitable to the synthesis of complex proteins. The mammalian cells possess the machinery to modify the proteins to the active form, post-translational modification, example like tissue plasminogen activator. The vectors, they are DNA molecules which can carry the foreign DNA fragment to the clone. These are self-replicating in an appropriate host cells. The most of important vectors or plasmids, the bacteriophage plasmids, or artificial chromosome vectors. The plasmids, bacteriophages, these are the cosmids. The cosmids are vectors of processing to characteristic growth plasmids on bacteriophages. This carries large fragments foreign DNA compared to the plasmids. The artificial chromosomes like bacterial artificial chrome and human artificial chrome, the yeast artificial chrome, these can be accepted large fragments for a foreign DNA. The methods for gene transformation and conjugation, the electroporation and liposomes direct to transfer to DNA. The applications of recombinant technique. What are the main applications? The manufacturers of proteins, hormones, and interferons, plasminogens, activating factors, and blood cutting factors, insulin growth factors, insulin growth hormones, acquired immunodeficiency deficiency syndrome test, and has been simply rapid, and diagnosis of molecular disease and sickle cell anemia, thalassemia, like familial hypercholesteremia and cystic fibrosis, and prenatal diagnosis like DNA from the cell collected from the amniotic fluids and chorionic village. The gene therapy, this is achieved by the cloning a gene into the vector that will be rapidly taken up, incorporated into the gene of host cell. Adenosine deaminase has been successfully treated. The applications of in agriculture, the genetically engineered plants are developed to resist the drought and disease. The good quality of food and increase the yields of crops is also possible. This is the industrial applications. In applications, it is also useful for the forensic science, transgenesis, gene cloning. That the cloning recombinant DNA molecules is a transfer of host cells within the host cell. It replicates the producing the dozens of the identical copies, and it is cloned with the cloned DNA can be recovered from the host cell, purified, analyzed, and transcribed. It is mRNA translated. Gene product is isolated and used for the research are sold commercially. This is structure and gene libraries. Collections of cloned DNA sequence. Each cloned segment relatively small. Many separative clones are required. Libraries, genomic chromosomes specific the complementary DNA, like information. This is also technically can be used for the retrieve any genes informations starting materials. Thank you. We are discussing about the recombinant DNA technology and what are the applications, what are the uses, and how in forensic science, how in the applications in companies, industrials, what are the uses for the recombinant technology, mechanism and procedure. Thank you, and thank you very much.